Welcome back to MLT 120. This is part 2 of the Anemias lecture. We're going to start off today by talking about thalassemias. Also, we're going to do some trivia questions along the way. So let's first start off by defining what thalassemia is. Thalassemia is a quantitative problem and it's genetic. It's a problem which there is a lack of production of the alpha or beta globin chains in our hemoglobin, and it impairs the ability to make either hemoglobin A, hemoglobin A2, or hemoglobin F, depending on the type of thalassemia that the patient has. So, this is different than the hemoglobinopathies that we talked about in part 1, because the hemoglobinopathies, there was just something wrong with the structure of the globin chains. One amino acid was exchanged for another amino acid on the chain, so there was a structure problem. So it was a qualitative problem. So with thalassemia, we've got a quantitative problem because we're completely lacking a chain. It's a lack of production of one of the chains. It's not just a structural problem with the chain. We have a lack of production with the chain. So that's the difference between our thalassemias and our hemoglobinopathies. So the morphology, the part that's written in red about the middle part of the slide here, the red blood cell morphology, we have red blood cells that are microcytic and hypochromic. So we have red blood cells that are smaller in size and that are lacking in color. We also have caused because of increase, destruction, and loss. This is because red blood cells will have abnormal morphology and shapes, will not live as long because the spleen removes it. Because remember, anything abnormal as far as red blood cells, if they're not that nice biconcave dish shape, the spleen's going to want to take them out. Thalassemia is more prevalent in certain populations of people. These include people of Mediterranean descent, Asian, Indian, Middle Eastern, North African, and Caribbean descent. Usually patients will present with our microcytic red blood cells or small red blood cells, but they will have normal iron levels in the blood. And we either classify thalassemias as either alpha thalassemias or beta thalassemias, and we'll talk more on slides coming up of the difference between the alpha thals and the beta thals. But it depends on the globin chains that are affected by the gene that's passed on in the patient, which they are, whether they're alpha or beta thal. And remember, thalassemia is genetic. Okay, remember how I just said on the previous slide that thalassemia is a genetic problem. Well, thalassemias, because it's a genetic problem, there's a problem with the genes in the thalassemia patient. So either there's a problem with the gene causing the gene to tell the globin chains in the hemoglobin to either be deleted, or there's a gene that's telling those globin chains to be defective. Okay, so let's look at the flow chart that we have down here. This model is actually showing how a hemoglobin chain or a hemoglobin molecule should be produced without a problem, without an issue like thalassemia. So let's go through that first, and then we'll go back through it and I'll tell you what happens in a case of thalassemia. So first we have our stem cell. And within our stem cell, we have our nucleus, and within our nucleus, we have these little X-shaped structures, which represent our chromosome. And our DNA is housed within our chromosomes, and our genes make up our DNA, and our genes tell our body what it's going to be, whether it's our globin chains, eye color, whatever. But in this case, we're talking about types of globin chains that we have in our hemoglobin molecule. So these genes are coding for different globin chains in our hemoglobin molecule. So this gene is going to code for the alpha globin chain. So it's going to code, and we're going to end up with an alpha globin chain. Okay. This one down here, this gene is going to code for the beta globin chain. So we're going to end up with a beta globin chain. So to make up the whole molecule for this particular molecule of hemoglobin, we're going to need 2 alphas and 2 betas. So this is the normal process of making a hemoglobin molecule. But in the case of a thalassemia patient, there's something wrong with these genes, or one of these genes. So let's say something is wrong with this beta globin gene, so it's going to end up not allowing this beta globin chain to be produced. So ultimately this beta globin chain is going to be deleted. So let's go ahead and mark this beta globin chain out so it wouldn't be produced. And if that beta globin chain is not produced, then we would not be able to produce our ultimate goal of a hemoglobin molecule, because you've got to have 2 betas and 2 alphas, or 4 total globin chains to produce a hemoglobin molecule. So if the gene is telling it to be deleted, or it's going to produce a defective globin chain, then the hemoglobin molecule cannot be produced. So that's why we end up with the cause of thalassemia being an increased destruction or loss, because we're not able to produce our hemoglobin as a result of our defective genes. So now let's compare and contrast alpha thalassemias and beta thalassemias, and we're going to talk about alpha thals first. Each person inherits 4 alpha genes, 2 from your mother and 2 from your father. And alpha thalassemias is a result of a gene deletion. So there's something wrong with the gene that's causing a deletion. And remember, you're inheriting a total of 4 alpha genes from your parents. So you can have situations where you can have 1 deletion of an alpha gene, or you can have 2, you can have 3, or you could have all 4 deleted. So let's look at those different scenarios down below, some examples. So the first example would be if you had all 4 alpha genes deleted. This is known as hemoglobin Bart's, or it's also called hydrops fetalis. And this usually results in stillborns of infants, or the infant dies shortly after birth within days. And again, this is if all 4 of the alpha genes are deleted. Okay, and it usually results in fetal demise and total body edema. Edema means swelling or excess of fluid. So Bart's kind of lines up with down here. Red is representing a deletion of alpha gene. So anytime on this little chart over here, if you see red, it means the gene has been deleted. So hydrops fetalis, all 4 genes deleted. Hemoglobin H disease is the deletion of 3 alpha genes. The patient lives with low hemoglobin levels, usually less than 10 grams per deciliter. Normal hemoglobin adult female is somewhere between 12 and 16, and a normal male hemoglobin is between 14 and 18. So again, this is 3. So you'll see here on the chart, representing in red, you have 3 of your alpha genes deleted. Some other symptoms might be splenomegaly. That means an enlarged spleen. If you have 2 genes deleted, this is known as alpha thal trait, or it may be referred to as minor thalassemia. Two genes are deleted, and you'll end up with mild anemia, and your red blood cells will be macrocytic hypochromic. Okay, and then you can also be a silent carrier where only one gene is deleted, and you usually have normal to slightly macrocytic red blood cells, and that's represented right here. And of course, your normal person without a alpha thalassemia would have all four alpha genes passed from their parents. Okay, time for a trivia question. Of the four clinical states of alpha thalassemia, which is incompatible for life or with life? A. Alpha thal silent carrier. B. Alpha thal trait. Hemoglobin H disease. Or D. Hydrops fatalis or Bart's. Okay, pause the video at this time and try to answer on your own before you move on to find the answer on the next slide. The answer is Hydrops fatalis or Bart's, hemoglobin Bart's. Let's talk about beta thalassemia now. This results from an inheritance of a defective beta gene from either one parent or from both parents. If you only get it from one parent, it's heterozygous. If you get it from both parents, it's homozygous. In the beta thal major, you can also refer to it as Cooley's anemia. We're talking about homozygous, where we've inherited the defective beta gene from both of our parents. You cannot make beta chains, so therefore we cannot make the hemoglobin A that we need. And it results in things like severe anemia, bony changes to the facial structure due to expansion of bone marrow and the bone marrow cavity, splenomegaly, which we've already mentioned of being an enlarged spleen, and failure to thrive by the ages of 2 to 4 years of age. And the majority of hemoglobin in beta thal patients is hemoglobin F because they cannot make that hemoglobin A. So remember, you have hemoglobin F as an infant, so that's usually the hemoglobin that you're going to have as a beta thalassemia patient, because you cannot produce that hemoglobin A. And you can see the image of the patient with Cooley's anemia or beta thalassemia major, and you can see how the facial bones and structure is a little bit deformed, and that's because of the expansion of the bone structure because of the bone marrow and the bone marrow cavities. In beta thalassemia minor, this is the heterozygous version where you just inherited the defective beta gene from one parent. Okay? And usually you have macrocytic hypochromic red blood cells. So you have small red blood cells that are lacking in color, and usually you have low or moderately low hemoglobin levels. But to sum up alpha thalassemia and beta thalassemia, it's a problem with the gene. The gene is either deleted or the gene is defective, so you cannot produce the globin chains that you need to produce in order to make the hemoglobin that you need for the body. Okay, trivia time again. The majority of hemoglobin in patients with Cooley's anemia or beta thal major is A. Hemoglobin A, B. Hemoglobin A2, C. Hemoglobin F, or D. Hemoglobin S. Pause the video before you move on and try to answer on your own. And the answer is C. Hemoglobin F. They still have their newborn hemoglobin F because they cannot produce hemoglobin A because their beta genes are defective. The next type of anemias we need to discuss are our hemolytic anemias. We're going to discuss each one of these on the slides coming up, but to list them out, our hemolytic anemias include our hereditary spherocytosis, our paroxysmal cold hemoglobinuria, or PCH, paroxysmal nocturia, hemoglobinuria, and the hemolytic disease of the newborn. These hemolytic anemias occur when our red blood cells lyse or are destroyed, thus making the cause an increase in destruction or loss anemia. Let's talk about hereditary spherocytosis first. It's more prevalent in Northern European ancestry. The red blood cell membrane has a problem. The spleen usually removes the red blood cell too soon because of that problem, and that was what contributes to the anemia. Most patients have the spleen removed, which is a procedure called a splenectomy, to lessen the degree of the anemia. So the cause is an increased destruction. The morphology is what we call a spherocyte, and it is microcytic, and it is actually hyperchromic. So we haven't seen a hyperchromic red blood cell yet. So this is an increase in color. So we have a small red blood cell that's increased in color. So if you look at the arrow in this image up here, this is a spherocyte. These are our normal red blood cells here, the normal size. So you see that the spherocyte is smaller and darker in color. But not all red blood cells in hereditary spherocytosis are going to be spherocytes. The test to diagnose a patient with hereditary spherocytosis is the osmotic fragility test. Hereditary spherocytosis patients' red blood cells will have an increased fragility when placed in tubes containing different levels of hypotonicity. So basically, the red blood cells will lyse sooner than a normal patient's red blood cells. So if you look at the image on the right-hand side of the osmotic fragility test, the top tubes, the line across the top, this is an example of a normal patient. And you can see the different percentages. It looks like we have 1%. The second tube has 0.85%. The second tube has 0.66%. The next tube has 0.5%. The next tube has 0.3%. And the last tube has 0%. So these have different concentrations of solutions that this sample has been diluted down with. So in the normal patient, we do not see any pinkish color until about the 0.66% tube. So red blood cells do not start to lyse until about this third tube. So this is for a normal patient. So when red blood cells lyse, they release their contents, their hemoglobin, and then you'll see that pinkish color in the solution. Now for this bottom line of tubes, this is for a patient with hereditary spherocytosis. Guess what? In this second tube, we're already seeing a pinkish color. So that's telling us these red blood cells are more fragile and they more easily lyse and release their contents. So that's telling us this is more likely or most likely a patient with hereditary spherocytosis based on this osmotic fragility test. Paroxysmal cold hemoglobinuria, or PCH, is characterized as increased destruction or loss as far as anemias. The red blood cell morphology is normocytic, normochromic. So we have a normal shaped red blood cell with normal coloring. As far as the anemia itself, it's usually rare. It's caused by the antibody P. And remember, antibodies are a blood protein that's part of the immune system. And antibody P is actually a cold reacting antibody. The antibody P attaches itself to the red blood cell and causes the red blood cell to lyse. The test that we usually use to diagnose PCH is the Donath-Lensteiner test. And usually what we do is we incubate a patient's red blood cell at 4 degrees Celsius which is considered cold, but that's actually within the range that we would keep our refrigerators at in the laboratory. And we also incubate at 37 degrees Celsius, which is our body temperature. And then we observe for hemolysis, or to see if the red blood cells have lysed. So if we look over at the image on the right-hand side of the test, these patient samples have been incubated. The first one was exposed to cold only, so at 4 degrees Celsius. 2B, the same sample was exposed at a cold temperature of 4 degrees and at the warm temperature of body temperature of 37 degrees. And then finally, the last tube, the patient sample, was exposed to warm only and no exposure to cold of 4 degrees Celsius. So we look for hemolysis. Of course, we have hemolysis, or the blood cells have lysed in tube A and tube B. So these tubes would prove that we are positive for PCH. We have a cold antibody. Next, we have paroxysmal nocturnal hemoglobinuria, or PNH. The cause is an increased destruction or loss anemia. Again, this falls under our hemolytic anemia category. The morphology is normocytic, normochromic. Again, this is a rare anemia. Occurs due to a problem in the maturation of the hemopoietic stem cell. Causes proteins to be missing from the red blood cells and hemolysis to occur. And the hemoglobin, when it comes out of the red blood cell as a result of hemolysis, that hemoglobin passes in the urine. It's called nocturnal because the red blood cells are destroyed while the patient sleeps or laying down. Due to the lack of proteins that help protect red blood cells, or the red blood cells form, they will more readily lyse, or hemolysis will happen. Tests used to diagnose PNH is the Ham's test and the sugar water test. For the Ham's test, it's also known as the acid hemolysis test. And what happens is acid serum is mixed with the red blood cells, and normal red blood cells do not lyse, but patients with PNH, their red blood cells will lyse. So that's what you're seeing here in about the middle of the page is the Ham's test. The C's represent the controls. PTs are representing the patient samples. RT is representing room temperature samples, and then the 37 is it was incubated at 37 degrees to replicate body temperature. So if you have samples that lyse, which we did in this sample here, that means the patient has PNH. Another test is that sugar water test. You mix the patient's red blood cells with a sugar water solution or sucrose, and you incubate the sample, and then you're going to observe for the degree of hemolysis. Again, you're going to mix your sample. Some use serum, that's your control, your sucrose, and then if it has hemolysis, it's positive for PNH. No hemolysis, then it's negative for PNH. Okay, we have a trivia question. Which of the following is the cause of hereditary spherocytosis? Is it A, the replacement of an amino acid on the globin chain? B, a genetic inability to produce globin chains? C, a red blood cell membrane defect? Or D, the genetic inability to produce alpha globin chains? Pause the video before you move on to get the answer. The answer is C, a red blood cell membrane defect. That's the cause of hereditary spherocytosis. Okay, here's another trivia question. Which of the following laboratory tests would be used to confirm PNH? Is it A, hemoglobin electrophoresis, B, the Ham's test, C, osmotic fragility test, or D, the Donath-Landsteiner test? Pause and answer before you move on. The correct answer is B, the Ham's test. It's also known as the acid hemolysis test or the acid serum test. The last hemolytic anemia we're going to discuss is the hemolytic disease of the newborn, or the HDN. This is due to the incompatibility of the Rh blood type between the baby and the mother. This is when the baby has a positive Rh type and the mother has a negative Rh type. The Rh factor is a type of protein found on the outside of the red blood cells and is genetically inherited and passed down from the parents. So if it's on the outside of the red blood cell, it's an antigen. And you'll learn more about that when you get to blood banking. But the situation is the mama delivers the first baby and is exposed to the baby's positive Rh. Therefore, this stimulates the mom to make antibodies against the positive red blood cells. Again, the mother is Rh negative. So the first baby will be born normal because the mother doesn't have any antibodies to start with. Okay? But because of that first baby, the mother's going to build up antibodies to Rh positive blood. The second baby and any future babies that have Rh positive blood will have their red blood cells attacked or lysed in utero or inside the uterus by the mother's antibodies because they are Rh positive and she's Rh negative. Because of this, the babies can become anemic. As the number of pregnancies increase for the mother's antibody titer will increase. Therefore, later babies may result in more severe anemia. And it may require even a transfusion in utero. Rh negative mothers are now given what we call a RhoGAM shot to prevent this from happening. The RhoGAM shot prevents mothers from developing those antibodies against Rh positive blood. If a mom did not receive prenatal care, may still see cases of hemolytic disease of a newborn. So in these cases, the morphology of the red blood cell is normocytic, normochromic. Okay, so normal size, normal colored. But the cause is increased destruction or loss. So if you look over here at the image, you see the baby, and the baby itself is positive, but the mother is negative. So the mother is going to produce, as a result of this baby being negative, the mother's going to want to produce antibodies against Rh positive blood. So to prevent that from happening, they're given this RhoGAM shot because we don't want our mothers to make antibodies against Rh positive blood because we don't want there to be problems for the second pregnancy and pregnancies after. The next category of anemias that we're going to discuss is our enzyme deficient anemias. And we're going to discuss two of those, our pyruvate kinase or PK and our G6PD. This occurs when patients genetically lack the ability to produce certain enzymes. And we're going to talk about each of these on the slides coming up. Our first enzyme deficient anemia is pyruvate kinase or PK. This one is a rare anemia, and red blood cells lack the enzyme pyruvate kinase or PK. They cannot generate ATP or energy from the Ebden-Meyerhof pathway, or that's also referred to as glycolysis. So you'll hear those terms used interchangeably. The EMF pathway and glycolysis are the same thing. So symptoms for PK include splenomegaly or large spleen, and that's what it's showing here. Your spleen should be normally about this size in the body, but someone with PK would have an enlarged spleen, and that's what it's showing here. A person with PK may be jaundiced. This is that yellow color to the skin. Even the whites of the eyes can be yellow in color. Another symptom is just overall fatigue, and that's where they cannot generate energy or ATP. We usually see this in our Amish populations, and a lot of times we don't see it a lot outside our Amish populations because it stays within that populations because they're not as genetically diverse. And the morphology is normocytic, normochromic as far as our red blood cells, and the cause is increased destruction and loss. The second enzyme deficient anemia that we're going to discuss is G6PD anemia. This is when a patient lacks the enzyme glucose 6-phosphate, and it's needed for the red blood cell metabolic pathway. When patients are exposed to some drugs or infection, we can see hemolysis of our red blood cells. The lack of the enzyme causes the red blood cell inclusion called Heinz bodies to form. And Heinz bodies are made up or composed of denatured hemoglobin. So you can see what a Heinz body would look like under the microscope in these images up here. So you have your red blood cells, and this little blue dot that's kind of hanging off of our red blood cells, these are Heinz bodies. Okay, so the spleen is going to want to remove the part of the red blood cell containing the Heinz body. So it results in the hemoglobin appearing as a bite cell. So this is the process that the spleen's going through. So these are the Heinz bodies. The spleen's going to want to go through and snip off that part of the red blood cell containing the Heinz body. So the red blood cell is going to end up looking like it's got a little bite taken out of it. So that's how we end up with a bite cell. And these are what bite cells look like under the microscope. So as a result of the red blood cells appearing as a bite cell, we're going to end up with hemolysis occurring. Okay, so the morphology of G6PD anemia is normocytic, normochromic, but the cause is going to be an increased destruction and loss anemia. We have a final trivia question for this lecture. The presence of Heinz body inclusions or bite cells noted in a red blood cell is indicative of which anemia? Is it pyruvate kinase anemia? Is it HDN? Is it PCH? Or is it G6PD anemia? Pause the video before you move on to see the answer. The answer, of course, is D, G6PD anemia. Those are the ones that you're going to end up with your Heinz bodies inclusions, and the spleen's going to want to take those Heinz bodies out, and you're going to end up with a bite cell. I hope you've learned a lot about the different anemias in this lecture, and please let me know if you need any help or need any clarification on anything. You may email me, you may come by, see me during my office hours, or you can see me before or after our lab time.